couple of, or at least one announcement, one of which is you got that too loud Ahern. I really appreciate the comments and feedback I got on what's working and what's not working for you. And one output of that, or one result of that, is I've decided to do an additional session. I will do this on a semi-regular basis outside of class, where I will help you to focus on what I've been talking about. The common thing that I'm hearing from people is that they're having difficulty sort of getting that bigger picture. And so I want you to have that, and I want to help you to focus your studying and your understanding. So my temporary solution to that is I will do an additional session with you that will likely occur on Thursday evening, early Thursday evening, okay? I haven't scheduled it yet, but I will inform you by email, and I will certainly let you know in class on Wednesday when and where that will be, okay? So I will videotape that, so if you can't make it for whatever reason, you'll be able to watch it on videotape, all right? And bring your questions, bring your comments, and I will have a sort of a short presentation that I will organize for that. So that was one thing. The second thing was the feedback about keynotes, or I use Keynote instead of PowerPoint, but the PowerPoints that you see and so forth. I feel that your feelings are like mine. I don't like PowerPoint. I don't like presentation software. So next term, my plan is to go back to the outline format that I've used before. You're seeing a sort of a midpoint of that right now. We'll sort of adapt to that or move in that direction the rest of this term. But next term, I will go back to my older format for that. So I think you will see with that, again, that bigger picture that I want you to see. So that's two changes that are coming. And if you have additional comments or suggestions or feedback, please let me know. Okay? Any comments before we dive in? Everybody's worn out from the weekend. Zombies. Zombies versus humans is done, right? Okay, well, let's talk then about, today will be the last, if I get through it, the last I'll be talking about the signaling process, okay? So last time when I started talking about the epidermal growth factor receptor, and I pointed out that that was different from the epidermal growth factor, and the epidermal growth factor, of course, is what binds to the epidermal growth factor receptor. The epidermal growth factor receptor is a receptor tyrosine kinase, RTK, that's found in the membrane of cells, and binding of the epidermal growth factor causes the receptor to dimerize, which starts that overall process like we talked about last time. This figure depicts the binding of, or actually depicts an epidermal growth factor that has already bound to the receptor, so it has already dimerized. You see the two dimers together holding that epidermal growth factor, which is shown in green. And autophosphorylation has already happened. That is, that inner portion where the tyrosine kinase is located has already phosphorylated each strand, so there are now phosphates on tyrosines on those inner portions of the EGFR. Okay? And you can see that those tyrosines have been a target for binding by the signaling complex. Those kinases can also phosphorylate other tyrosines on other proteins. And you see one, GERB2, for example, that has a phosphate attached to it. Okay? Everybody understand? Now, the important thing I want you to take away from this image, and I'm going to show you the signaling process goes further, all right, along. But the important thing I want you to take away from this is that protein at the bottom called RASS. RASS is really critical, as we're going to see. It's going to be critical in the first part of the lecture, and it will be critical later in the lecture as well. RASS is a very, very interesting protein. It plays central roles in the cell's decision of whether or not to divide. Now, RASS is what we describe as like a G protein. It's not a G protein, but it behaves like, I shouldn't say it's not, it actually technically, I guess, is in some sense, but it behaves like the alpha subunit of the G protein that we've been talking about. That is, it binds to guanine nucleotides. And you can see that the RASS here has been bound by a GDP, and that the assembly of this signaling complex has caused it to let go of that GDP and bind to a GTP, just like the alpha subunit of the G proteins that we've talked about before. Now, you might expect that that binding of the GTP would activate RASS, just like it activated the alpha subunit of the G proteins, and in fact, you would be right. It does activate it. So, what we've seen now is the binding of EGF by the EGFR has started a process that has resulted in the activation of RASS. Okay? There are several RASS proteins we have in our cells. We won't talk about individual ones, but we have several RASS proteins and RASS-like proteins that play very important roles in the processes I've talked about with respect to cell division. Okay, so there's RASS bound to GTP. Now, I told you that the signaling complexes are the signaling process is fairly complicated for these RTKs, and we can see that happening right here. Yes, question? RASS is a protein that we describe. His question is, what is RASS exactly? RASS is a protein that plays a role in the signaling process, and it's what we describe as a proto-oncogene. I'll explain that later in the lecture. Okay? But for our purposes right now, it's simply a protein that plays a role in the signaling process. All of these are proteins. Everything that you see on the screen here is a protein. Okay? RASS binds to a protein called RAF. Yes? Yes. RASS joins the signaling complex and gets activated by that signaling complex. Yes. So does the activation cause it to bind? The activation causes it to become activated. Just like we saw in the beta-adrenergic receptor, where the binding of the hormone caused the alpha subunit to become activated, that was to get rid of, let go of its GDP and bind GTP. Similarly, the signaling complex that was shown in the last slide causes RASS to let go of GDP, bind GTP, and become activated. Now, you see that what's happening in the rest of this signaling process is it's not all occurring in one place. RASS goes and travels to bind to another protein called RAF. And by the way, I'm not asking you to memorize the proteins here. All right? The protein that I'm talking about, RAS, is one that you should understand, and you should know how it's activated. It's activated by the epidermal growth factor receptor, by the signaling complex there, but I'm not expecting you to know individual protein names. Okay? RAS you should know. All right, so RAF gets activated. We see phosphorylation of another protein called MEK. We see phosphorylation of another protein, which in turn phosphorylates another protein called MAPK, which phosphorylates several proteins. And we see this signaling cascade like other cascades we've seen inside of cells. You remember that cascades cause things to happen quickly, and they also cause signals to be amplified quickly. So this binding of this epidermal growth factor has stimulated a process that's really changing the direction of this cell pretty quickly and without question. That's happening, okay, so that the cell will go through and divide. That's what's all happening here. That's what the upshot of all of this ultimately is. Now you'll notice that all three of those proteins at the bottom of the figure point into the nucleus and say transcription, meaning that this whole signaling process is aimed at stimulating transcription of genes that will result in DNA replication and cell division. Okay? The bottom things that are happening here are stimulating transcription of genes that will stimulate DNA replication and cell division. At that point, the information has been communicated and the cell has responded accordingly. Everybody got that? Good. Okay. there, 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 and ultimately we're at the bottom. Okay, well, let's talk a little bit about RASS. RASS is an interesting protein. It's a small protein. It's not at all unlike the alpha subunit of the G protein that we've been talking about. And as I said, there are multiple RASs. It is actually a family of related proteins. Each one of these is monomeric, meaning only a single subunit, and behaves like the alpha subunit of G proteins. Like the alpha subunit of G proteins, RASS binds to guanine nucleotides. You saw that it was bound to a GDP, which left it in the inactive state. And that activation of it resulted in the letting go of the GDP to bind GTP, which put it in the active state. So when it's in the active state, that signaling process on the last slide occurs. Okay? There's a depiction of human RAS, which is bound to GDP. And I'm going to say more about RAS near the end of the lecture where I talk about oncogenes. Okay? Like the alpha subunit of the G proteins, RAS is a GTPase, meaning it converts GTP into GDP. It does that inefficiently, just like the alpha subunit of G proteins does. And it also means that RAS is able to turn itself off. And that's really important because if we think about it, we don't want RAS to be active all the time, because if it is active all the time, that cell is going to be continually getting this signal, divide, 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 and we know what that leads to. And we'll see how that leads to problems later. Okay. Now, I've made a summary. I'm not going to read through all those. I've made a summary of what I've been talking about with respect to the RTKs. Okay? That may help you to get the major points of the things that I've talked about, and I put it there for that purpose, obviously to help you to understand the major parts of RTK signaling. All right, let's talk about another type of signaling. Another type of signaling is steroid hormone signaling. Okay? Steroid hormone signaling, as I mentioned earlier, is different from membrane signaling, and the reason that that's the case is because steroid hormones have the ability to cross the lipid bilayer, and when they cross the lipid bilayer, they can exert their effects directly inside of cells. They do not require a membrane protein. The RTKs all required a membrane receptor protein. Okay? Or the RTKs actually all were membrane receptor proteins because their hormone could not cross the lipid bilayer. Okay? Their hormone could not cross the lipid bilayer. So if they can't cross the lipid bilayer, they darn sure better be located in the membrane so they can pick up that signal. If they can't pick up that signal, they're not going to do any good. That's not the case with steroid hormones. They cross the lipid bilayer reasonably easily. Now, I've shown on the screen here some steroid and other related hormones that you should memorize the structure for the next exam. All right, people are smiling good. I'm not going to expect you to memorize the structure of those, right? Oh, collective sigh of relief, right? God, he wouldn't do that to us, would he? But they're important. You should memorize them, right? I mean, how about that calcitriol? That's the active form of vitamin D, right? No. Nobody's going to pay attention after that. All right. But no, the structures aren't important. But you can see some things up here. You see the standard steroid hormones, estradiol, female sex hormone, testosterone, cortisol. Okay, we've talked about actually cortisol already a little bit. Vitamin A. Vitamin A can also act like steroid hormones. We see the thyroid hormone receptor, thyroid hormone, which binds to the thyroid hormone receptor. Thyroid hormone, I think I mentioned last time, is the only molecule in the cell that I'm aware of that actually is covalently bound to iodine. And, of course, vitamin D receptor. Vitamin D, which binds the vitamin D receptor. Monday morning. Okay, so steroid hormones are important for a variety of physiological purposes. All right? They help to control metabolism. If I said, how does any hormone control metabolism, what would you say? There's two ways. How would a hormone control metabolism? I'm getting the silent treatment again. Yes? So she's asking about if adrenaline can slow your metabolism, and adrenaline affects your metabolism, certainly, and it has different effects on different cells. So I won't give a standard answer to that question, no. But I'm wondering how, based on the mechanisms that we've talked about in class, would a steroid hormone affect metabolism? Should I just save that for the exam? Nobody wants to take a shot at that? What are the mechanisms for controlling something in the cell? Question, comment? Yeah. Is it through allosteric regulation? Okay. 1 point extra credit right there. Oh, yeah, it pays to talk, doesn't it? Okay. See me after class. I'll make sure you get the extra credit, okay? Now, anybody want to take another shot? Yes? Gene expression, absolutely. The two mechanisms that cells have that we've talked about for controlling metabolic pathways are allosteric regulation, where you're affecting the enzyme, and gene expression. Another point there. See me after class, okay? Now, these are ways that cells have to do that. How can we affect things allosterically? All right? Or more importantly, more broadly, by controlling the enzymes. Allosteric regulation is part of that, as we will see. What's another way of controlling enzymes besides allosterism? Yeah. Competitive non-competitive inhibition. Not really. That's sort of a derivative of allosterism anyway, but nice try. Maddie? Be more specific. She said proteases. What is a protease causing to happen in a protein? What does a cut mean? What if I put a phosphate on? Would that affect things? Yeah? Yeah, it could. So there's a broader category I'm looking for here. Who wants to try the broader category? Yeah. Well, phosphorylation, but it doesn't have to be phosphorylation. She said it could be protease. I'm thinking of the broad category that encompasses those. Yeah. Covalent modification. Thank you. Extra credit point right there. Okay? Covalent modification. That's how it is that enzymes get controlled. Covalent modification and allosterism. And the third is by whether or not they're made. That's why the gene expression is important. Okay? Okay. So, as I said, steroid signaling involves intracellular receptors, not membrane receptors, but intracellular receptors. And there are 5 classes of steroid hormones. We'll actually talk about those next term, not this term. All right? And the signaling mostly affects gene expression. Mostly affects gene expression. We'll see how that happens in just in a second. So because of that, when we see things that affect gene expression, the result is a slower response. Changes in gene expression in eukaryotic cells can take hours or days in some cases to become active. That's really important, okay? If I am running away from a grizzly bear, as we have talked about, if I'm running away from a grizzly bear, I don't want that response to take hours to days to happen. Gene expression would not be a good way to activate that. What would? Yeah. Like a... So yeah, the covalent modification that we've talked about, right? So having something or some sort of enzyme control, allosterism or covalent modification, happens much more quickly. And we'll see that happens when I talk about glycogen metabolism. Okay? All right. Let's look at steroid hormone signaling. You've already seen this figure once. Okay? Steroid hormone signaling happens when the steroid hormone crosses the lipid bilayer. We don't know exactly how that happens. We don't know exactly how that happens. I lied a little bit in that things like vitamin D actually use a protein to help them get across. But many steroid hormones simply cross all on their own. So for our purposes, we will consider steroid hormones all to be able to just cross the membrane on their own. After they cross the membrane, they encounter their receptor. And that receptor may dimerize, just like we saw the receptor tyrosine kinases do. We'll see that in a second. Okay? The receptor can be located in the cytoplasm. The receptor can be located, in some cases, in the nucleus, which would mean that the hormone would need to travel into the nucleus as well. Suffice it to say that the binding of the receptor, binding of the hormone by the receptor causes the receptor to change. And we've seen over and over and over now this term. Binding of something by a protein causes a slight change in the protein, and that slight change in the protein is necessary for the signaling process to occur. I've shown it here as binding of a hormone causes the receptor and the hormone to be taken into the nucleus, where they affect transcription. This turns out to be a fairly simple process compared to what you've seen with the RTKs. This happens fairly simply. Hormone makes it in, binds to a receptor, the complex makes it to the nucleus, and transcription is affected. Everybody with me? Okay. There we are. There's our hormone in the blood, crossing of the lipid bilayer all by itself, binding the receptor either in the cytoplasm or in the nucleus. Okay. That change in shape on binding causes the signaling process now to occur. Transcription factor, and by the way, a transcription factor is simply a protein that affects transcription. That's all it is. Okay? Protein that affects transcription. That transcription factor bound to the hormone then alters the cell's gene expression. So that means that genes that were previously not being transcribed now might be transcribed, and those that are being transcribed, we might stop their transcription. So in some way we're affecting transcription. Yes? I didn't hear the last part of what you said. Yes. Oh, good question. Okay. So the question is it true that it's not until the receptor is bound to the steroid that it's able to traverse this path that you see and make it into the nucleus? That's true for any of the receptors that are in the cytoplasm. That's correct. And what it's depicting, that sort of entry path there, that little tunnel it's traveling through, is that the nucleus actually is also a barrier, and it's a different kind of a barrier. It has what are called pores that regulate movement into and out of the nucleus. So we're actually seeing it traveling through a pore. But in response to your question, yes, that's only happening after it is bound to the steroid hormone. Yeah, another question. Yeah. Doesn't the receptor protein have to have a transport protein to make it across? In many cases, yes. Okay, but it depends, and so I'm keeping this simple for you for the overall view. But yes. Okay. Well, let's look at that process happening in a typical cell. We look at this. We see there's our cell with a lipid bilayer. We see it has a nucleus, and we see that we have a receptor protein in the cytoplasm. In this case, the receptor protein is bound to another protein called HSP70. HSP70 is called heat shock protein 70. For our purposes, it's just a protein. Binding of the HSP70 by the receptor protein keeps the receptor protein from being able to go to the nucleus. It's keeping it in the cytoplasm. As long as the receptor protein is bound to HSP70, it's going to stay in the cytoplasm awaiting a signal to move in. The hormone arrives in the blood outside the cell, and then the hormone by itself moves across the membrane. Okay? The hormone is now in the cytoplasm of the cell, and in the cytoplasm of the cell, it sees a receptor, and it binds to it. And the binding of the hormone by the receptor causes the hormone to change shape. Again, we see this over and over. The change in shape causes it to let go of HSP70. Well, since HSP70 is what was keeping it in the cytoplasm and keeping it out of the nucleus, what we see then is movement of the receptor with the hormone into the nucleus. Once in the nucleus, transcription gets stimulated because this is a transcription factor that is able to start the process of transcription. Yes, question? Where are the receptors in the cell usually located? Well, it depends, again, on the type of receptor that we're talking about. RTKs, of course, are found in the membrane of cells. Many hormone receptors are found in the membrane of cells. The beta-adrenergic receptor was found in the membrane of cells. Steroid hormone receptors are typically found, as I said earlier, in either the cytoplasm or the nucleus, more commonly in the cytoplasm. Okay. Here's a complicated thing I'm not going to ask you to reproduce. Yes? What happens to the steroid hormone? That's a very good question. What happens to the steroid hormone after it's done doing its job? Because I've made the point to you already that if you have a signal turned on, you've also got to have a signal turned off, right? So the cell has a mechanism that is more complicated than we're going to be able to go through here, but has a mechanism of turning that signal off. And when that signal gets turned off, that steroid hormone receptor can get recycled right back out into the cytoplasm, bind to an HSP70, and start that whole process over again. But that's a little bit more complicated than we can go through here. All right, so here's the typical signaling for a glucocorticoid hormone. That's a steroid hormone. We see exactly the same steps I've already shown to you in general. First of all, we see the hormone entry into the cell. We see the release of a heat shock protein. That's the HSP that's there. We see the binding and the dimerization of the receptor inside the cytoplasm. That dimer now makes it into the nucleus by going through a nuclear pore, as I mentioned earlier. And we see transcription being activated, exactly as we talked about in the general scheme before. So the binding of a glucocorticoid hormone is going to have some effects on this cell. Again, they're going to be slower effects than we would see if we had enzyme modification in some way. Questions here? Yeah. Does the dimerization happen in all hormones? No, it doesn't. But since I mentioned dimerization, I wanted to show you one here where dimerization actually occurs also with steroid hormones. Dimerization happens almost always with the membrane RTKs. The exception was insulin, which was already a dimer, okay? But that's the sort of lone exception. The dimerization happens in the membrane of cells with RTKs, and that almost always happens. Did I see a hand? Or here, I see a notice of a hand. Yes? There are two parts, yes? Yeah, that's sort of a schematic representation, so I don't want to say that's an absolute. You could also see, like you've seen before, one being between the two, so don't read too much into seeing two hormones on there. Okay. Hand, yes. I'm sorry, say it again. Does release of HSP70 cause dimerization? Not necessarily. So not every one of these is going to dimerize, as I said. So release of HSP70 is important for the molecule getting the signal to go into the nucleus, but not necessarily for dimerization. Okay? Good. Okay. So we've been talking about hormone signaling, and I've mentioned to you also that there's other ways that cells can talk to each other, and you know that as well. Okay? So Nerve transmission is a prime example. One part of the body talking to another part of the body. Our brain saying, contract that muscle, okay? And that's a signaling process that is almost instantaneous. Happens in milliseconds in many cases, thousandths of a second. And as I said also, I'll be talking about nerve transmission next term, not this term. But I, again, want to keep that on your radar, that that is another way of signaling to occur. Nerve transmission works as rapidly as it does because it relies on ion gradients. An ion, of course, is a charged molecule. And a gradient is a difference in concentration. So nerve transmission works because if you open up gates and ions rapidly flow in, they flow in very rapidly, cause a voltage change, and it's literally like an electrical circuit that's changing. And electrical circuits change very, very rapidly. Okay? Okay. Since these types of gradients and this type of ion movement requires the movement through ion channels, like I talked about previously, right? If we can block those signals by ion channel blocking molecules. And some of these molecules are actually neurotoxins. Some of the things that will block these are neurotoxins, meaning they're toxic to nerve cells because they're blocking that movement and causing some real problems. And again, we'll talk about those next term. Another group of molecules that's involved in signaling are called prostanoids, or as we used to call them, prostaglandins. Prostanoids act like hormones, but they're not hormones. They only act in the local region around where they're made. They do a variety of things, okay? This includes things like pain, okay? They stimulate uterine contraction. They can affect blood pressure. They do many, many things, and we will also, again, talk a little bit more about those next term. But I want you to be aware of them because they do behave like hormones. Now, a really good example of a pain set of prostaglandins is stimulated by when you get stung by a bee. You get stung by a bee, right? The cells around the bee sting release a protein and synthesize prostaglandins that result in swelling and a lot of pain. Okay? And that's happening localized to the area where the bee sting occurred. All right? You get stung by a bee on your arm, okay? You don't feel the pain in your head. Right? It happens only at that localized site. Now, you do sometimes feel pain in your head from prostaglandins as well, but it's in your head and not in your arm, right? So it's localized to the place where the prostaglandin is being made. Now, notably, prostaglandins synthesis is inhibited by molecules like aspirin. Aspirin inhibits the synthesis of prostaglandins. And since prostaglandins are involved in some of the pain responses, you take aspirin to deal with a headache because you're inhibiting the synthesis of prostaglandins that are causing that headache in the first place. Prostaglandins are precursors of molecules called thromboxanes. We talked about thromboxanes with respect to what process? Blood clotting, right? Okay? And so I said that this was a way of what we call thinning, or doing some blood thinning. It's not really thinning it, but we're reducing the ability of the blood to clot by inhibiting the production of thromboxanes. And we're inhibiting the production of thromboxanes because aspirin is inhibiting the production of prostaglandins in the first place, which are the precursors of the thromboxanes. Okay. And I see I need to scroll this up a bit, don't I? Okay, just a second. There we go. Yeah. Okay, good question. So does the synthesis of prostaglandins necessarily result in production of thromboxanes? The answer is no. Okay? No. So it can, but if we slow down the synthesis of prostaglandins, we will slow the production of thromboxanes. That's really the thinking in terms of taking aspirin to reduce the incidence of clotting. Okay? Is this a question?
Yes.
Prostaglandins, or question is, are they transmitted through the blood? They can actually travel a little bit in the blood, but it turns out the reason that prostaglandins work so locally is they're not very stable. They get broken down pretty readily by the body. Okay? Okay? Okay. And synthesis inhibited. And oh, by the way, they can also be inhibited by steroids. We use steroids sometimes to treat swelling. If you have real bad swelling, they will give you steroids to stop that. And that's because prostaglandins are also involved in the production of material that causes swelling. Okay? Inflammation can be stimulated by prostaglandins. The way that the steroids work is different from the way that the aspirin works. We'll talk about that next term. And steroids, we don't want to just take in general, so we want to be careful how many steroids that we are taking. And so we will usually only give steroids to treat swelling if there's a very severe type of response. Yes?
So for someone like with swelling in the vertebrae, when they get like a cortisone shot, is that what this is?
So when a person gets swelling in the vertebrae and they get a cortisone shot, is that what this is? And this is certainly affecting that because it's, again, going to affect the local production of the prostaglandins there, which may be involved in both pain and swelling. Absolutely. Yep. Cortisone is commonly what's injected into the spinal, near the spinal cord to reduce back pain. Yeah. Yep. 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 So asthma medications contain steroids. Some of them do, not all of them do, but some of them contain steroids. Is that something you want to be doing every day? Well, again, I'm not giving medical advice, so I'm not allowed to do that. But I will tell you this. I'm an asthmatic. I take inhalers. I bet that's where you get the steroids typically. You're getting them in there. And those are not considered to be too bad because they go to your lungs and they're acted on pretty quickly. They don't travel in the rest of your body. So it is felt, at least I know from talking to my doctor, that taking inhalers is better than the alternative, which is the wheezing and so forth that's associated with asthma. And it's wheezing in asthma that can lead to emphysema, actually. So it's considered less problematic for you to be taking in steroids in an inhaler, at least by my doctor, than otherwise. So again, consult with your doctor for individual advice. If my lawyer says that, right? Yes? Glucocorticoids, uh-huh.
Yeah, how does like an inhaler with steroids act so quickly?
So how does an inhaler work so quickly? Well, that's a little bit more, that's getting off the subject of what I'll talk about here, but I'll briefly answer your question. Inhalers, what they're doing is they're breaking down some of that congestion in your lungs, which is favoring that wheezing process, and that is an immediate response because it's acting directly on the site. But that's a little bit different kind of phenomenon than what I'll talk about here. That's not, as you might expect, that is not a gene expression phenomenon. There are rapid-working inhalers, and there are slower-working inhalers. So slower-working inhalers might, in fact, be more involved in gene expression. So it's not an absolute one or the other. Okay. Well, let's talk about what I like to call signaling gone wild. All right? Signaling gone wild is what happens when we have problems that are happening in the signaling process. Most often those problems happen as a result of mutation. Okay? Signaling proteins, I hope I've convinced you, play very important roles in controlling the overall decision-making process of a cell. They affect the cell's metabolism. They affect the cell's decision to divide, and that's the two of the most important things that cells do. I want to give you some definition. An oncogene, we commonly hear that term. Oncogene is a protein, it's a cellular protein, that has gotten mutated, and that mutation leads to uncontrolled growth. Notice that the term oncogene is a mutated protein. Okay? The unmutated protein has a name of its own. It's called a proto-oncogene. So, we have these normal genes floating around in our cells that control processes in signaling. If they're not, we're in trouble. So we want to have proto-oncogenes. We want them controlling the cell's decisions. It's only when they get mutated or something related to them gets mutated that we have a problem. It's the mutation, the mutated form that we call the oncogene. Proto-oncogene, unmutated, oncogene, mutated. All right? Now, mutations in signaling systems can lead to tumor formation because they can favor uncontrolled growth. We'll see a couple ways in which that happens briefly. All right? Those mutations may affect protein structure and function. They may also affect the amount of a protein that's being made. We'll see examples of both of those with respect to oncogenes. There may be other mutations as well, and I won't talk about other mutations. All right, let's start with RASS. RASS is a proto-oncogene. Mutation of RASS leads to formation of an oncogene. RASS is really critical in this process. Okay? RASS, as we saw, binds to guanine nucleotides. When it's bound to a GDP, it's in the inactive form. When it gets loaded with a GTP, it's active, which means it's going to stimulate cellular division. And that RASS has a way of turning itself off. It's a GTPase, meaning it cleaves GTP to leave GDP. Okay? Here's the scary part about RASS. RASS, mutations in two different parts of RASS can lead to inhibition of the GTPase. Now, when that happens, RASS is no longer able to turn itself off. RASS binds to GTP, and it's on, and it says divide, 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 and it forever says divide. We can imagine that that's problematic. It turns out that mutations in RASS that affect any of those amino acids at those places result in a RAS you can't turn off. Scary, guys. When you want to think about pollution, your air, your water, the crap you eat in your food, right? That's what causes mutation. That's why people worry about these things. Here's a single mutation in a single protein that can have a drastic effect on your life. Okay? Mutated RAS is the most common point mutation meaning a single base being changed. It accounts for, it's found in 90% of pancreatic cancers and in 20% of all cancers. This is a very important protein. We don't want to mutate RAS. Okay. I've talked about receptor tyrosine kinases. However, not all receptor tyrosine kinases, or not all tyrosine kinases are RTKs. That is, there are tyrosine kinases that are not found in membranes. Okay? They're not receptors, but they are tyrosine kinases. Good example, protein called SRC. SRC was actually the first oncogene that was ever discovered. And this may surprise you, but it was discovered over 100 years ago. Over 100 years ago, in chickens. Yeah, question. Do we have drugs that can inhibit RAS? We probably would not want to use them, right? Because RAS is a normal protein that's found in our cells, and we don't want to muck with that. So RAS is not a good target for drugs. Okay. All right. SRC proteins are tyrosine kinases. They were found in chickens 100 years ago because they found that a virus in chickens could cause cancer. It's called Wrasse sarcomavirus, R-O-U-S, sarcomavirus. And the SRC comes from that name, sarcomavirus. This virus was carrying a mutated form of a chicken's cellular SRC. And that mutated form was out of control and causing cells to divide uncontrollably. Now, SRC is controlled by phosphorylation. In the normal form, it's dephosphorylated and stimulates cell division. If it's phosphorylated, its signaling is turned off. So if you don't want this cell to divide, you would phosphorylate SRC. However, if mutation affects the ability to phosphorylate it, what's going to happen to SRC? It's going to be left on. And that's what was in this virus. This virus couldn't be phosphorylated, and so the SRC that the virus was injecting into the cell was causing the cell to become cancerous and divide uncontrollably. And it was within a virus, so it was infectious. Most cancers are not infectious. Okay? All right. How am I doing on time? 12:46. All right. Sarc is an example of a protein, of an oncogene, whose function / structure is affected by the mutation. It's affected by the mutation. In this case, the cell is unable to phosphorylate it, is unable to turn it off, it's left in the on state, the cell's got a problem. Okay? The thing that gets phosphorylated in Sarc are tyrosines. If you mutate Sarc so that you lose those tyrosines, you're left with the rest of the protein, which can completely signal and cause cells to divide. These tyrosines are places you don't want to mutate, folks, because you want to be able to turn this off so that it functions properly. And if you mutate those, you make the oncogene, which now causes uncontrolled division. It can't be turned off. Okay? All right. I'm almost out of time, so I'll save this for next time, but I do have a song I hope you will enjoy. Please join me. It's called Student Nightmares. Okay? I answered 3B, but then I thought it might be C. Or was the false true? I can't undo, it makes me blue. Asked me to list all the enzymes that regulate fat. As I wrote them down, I discovered I didn't know jack. I ought to give thanks, scoring some points, filling in blanks. I squirmed in my seat, feeling the heat, shuffling my feet. Professor, jump the gun. Professor then told me there wasn't a chance I would pass. So I started crying and fell through a big pane of glass. I suffered no harm, 'cause I awoke to my alarm. Oh, nothing compares to deadly scares of student nightmares. I still have those dreams myself, guys. Okay.